Hello, I'm Dr. Mark Darcy, and today I'm going to give a lecture on Mendelian genetics, an area of biology that we should all at least try to develop some familiarity with. So let's get started. Okay, Mendelian genetics with me, Dr. Mark Darcy. So let's start with an introduction to genetics. Genetics is a branch of biology that deals with heredity and variation of organisms. So on the right, you can see a chromosome, a sort of stylized version of a chromosome anyway. Now, chromosomes carry the hereditary information, the genes. So a certain chromosome, for example, chromosome 1, would carry a certain set of genes. Chromosome 2 would carry a different set of genes. All of our genes come in pairs because we have two sets of chromosomes, one set from our mother and one from our father. So two chromosome 1s, two 2s, 2, 3, 4, 5, etc. There are 23 pairs. Now, chromosomes carry the genes. The genes are basically just a region of the chromosome with a particular sequence of DNA, a particular sequence of nucleic acids. The arrangements of these nucleotides, adenine, thymine, guanine, and cytosine, A binding with T, G binding with C, make up our genes, and they can be thousands and thousands of bases long. We have about 2 meters of DNA in each one of our cells, and we have a lot of nucleotides, and they make up genes, and the estimates vary, but we think we have about 25,000 different genes, each coding for a different protein. The way it works is DNA gives rise to something called RNA, and RNA goes to a machine, an organelle in our cells known as a ribosome, which then makes proteins. So the gene carries the instructions to make a particular protein. The RNA carries those instructions from the DNA out of the nucleus to a ribosome in the cytoplasm of the cell, and the ribosome makes a protein. For example, there might be a gene that codes for making insulin. When that gene is active, when it is switched on, then messenger RNA will be made from that particular gene. That RNA will go to a ribosome, the ribosome will read it, and insulin, the protein, will be manufactured. So we have lots of different genes, and they all interact with each other and allow the organism to form the way it does. Now, there's something known as the central dogma of molecular biology. It was formulated by Francis Crick, who together with James Watson worked out the structure of DNA. I should also mention that Rosalind Franklin was key to that discovery. She performed some X-ray crystallography experiments which helped to discover the structure of DNA. Okay, so central dogma of molecular biology is DNA makes RNA makes protein. Okay, so chromosomes and genes occur in pairs. Pairs of genes exist on homologous chromosomes. So two chromosome 1s carry the same genes, two chromosome 2s, 3s, 4s, 5s, etc. They're known as homologous chromosomes. New combinations of genes occur in sexual reproduction, fertilization from two parents. The reason for this is that you get one set of your chromosomes from your mother, one from your father. But when you produce sperm or eggs, depending on whether you're male or female, only one set of your chromosomes is deposited in a particular sperm or a particular chromosome. So only one set is passed on to the next generation, where it combines with the set of the other parent to make a new offspring that is a combination of both parents. So homologous chromosomes, for example, two chromosome 1s will carry homologous genes. Okay, so further reminder, genes. Genes are areas of DNA which specify the synthesis of particular proteins. Alleles, most genes can come in more than one variety. These versions are called alleles. So a diploid cell, that is a cell with two sets of chromosomes, one from each parent, could have identical alleles. So for example, perhaps there is a gene that codes for eye color. Now, you could have a blue-eyed allele from one parent and a brown-eyed allele from the other. Or you could have two blue-eyed alleles. So you could you could have different versions of the same gene. These are known as different alleles of the same gene. Okay, so in an haploid gamete, so diploid is when you have two sets of chromosomes, haploid is when you have only one set, eggs and sperms only have one set. So in a gamete, in egg or a sperm, there will only be one of the two possible alleles. So only pass on one of your alleles to the next generation. Homozygous and heterozygous. So individuals with the same allele on both chromosomes are homozygous. With different alleles, they're heterozygous. So two blue-eyed alleles of a gene would be homozygous. A blue-eyed and a brown-eyed allele would be heterozygous. The genotype is a total collection of genes in an organism. The phenotype is the external appearance of the organism, which results from the genotype. So genes carry instructions, but the result of those instructions is known as the phenotype. The sequence of the DNA is the genotype. The outward behavior, the phenotype of the organism is determined by the genes, but also by the environment. So there is genotype, the sequence of DNA, and phenotype, whatever outward trait is expressed. A cross, when two different individuals mate, they each contribute one copy of each chromosome. When it is organized for scientific purposes, this union of gametes is called a cross. A self-cross, when two individuals which are genetically identical are mated, this is called a self-cross. So if you take two people with blue-eyed, if you take two individuals with one trait, so they're bred to have one particular trait. So for example, a plant with long stem or with blue flowers, and you basically you can mate those together and you will get an offspring. Now, if you mate an organism with itself, this is known as a self-cross. It's not uncommon in haploid organisms or in plants, but it's rare in animals. A zygote is the individual produced from the creation of a diploid fusion of two haploid gametes. So when, for example, an egg and a sperm fuse, you get a zygote, which then divides and divides and divides to make the organism. So as we can see here, we've got the chromosome. We have got the chromosome, which when you zoom in, you can see genes. And you could, on homologous chromosomes, have different alleles of a particular gene. So now let's talk about man himself, Gregor Johann Mendel, the person who came up with Mendelian Genetics. So this is the man right here, Gregor Mendel. He was an Austrian monk born in what is now the Czech Republic in 1822. Son of a peasant farmer, studied theology and was ordained a priest in the order of St. Augustine. Went to the University of Vienna, where he studied botany and learned the scientific method. He worked with pure lines of peace for 8 years. Prior to Mendel, hereditary was regarded as a kind of blending. So basically the two parents would blend together to make a child that was a mixture of the two. So basically the idea then wasn't that there were these separate alleles of genes, a blue-eyed and a brown-eyed allele, for example, but that there was a sort of merging each generation of the parent trait into one mix of the two. So a sort of fused allele. That was what was believed until Gregor Mendel came along. So Mendel's peas. Mendel looked at 7 traits or characteristics of pea plants. And these are those. So basically, were the peas round or wrinkled? Were they yellow or green? Were the flowers purple or white? Inflated or pinched ripe pods? Green or yellow unripe pods? Axial or terminal flowers? Long or short stems? So he looked at purebred versions of the plants that had got one of these 2 traits. So in 1866, he published experiments in plant hybridization. I'm not going to try and pronounce the German version of it, in which he established his 3 principles of inheritance. He tried to repeat his work in another plant, but it didn't work because the plants reproduced asexually. So they basically just budded and produced copies of themselves. So they didn't have this merging of different traits, these different alleles in the same way that plants that reproduce sexually do. If, going back to if he'd tried the second plant first, then he may not have come up with his ideas. So it's lucky that he did try a plant that reproduced sexually. His work was largely ignored for 34 years until 1900, when three independent botanists rediscovered his work. Another kind of, this sort of happens quite often in science. Someone will discover some process, come up with some idea, but if it's not publicized, then the work can be forgotten about and maybe only later rediscovered independently. It's quite a shame that Mendel's work wasn't, first of all, published in a language other than German to be more widespread, and that it didn't become popular, because at the time of Mendel, there was also Charles Darwin, and Charles Darwin, it seems, was unaware of Mendel's work. And if he was aware of Mendel's work, it may have actually helped. Darwin to beef up some of his ideas. I mean, they had a bit of beef anyway, but a bit more beef couldn't have done any harm. Okay, so Mendel was the first biologist to use mathematics to explain his results quantitatively. Mendel predicted the concept of genes, that genes occur in pairs, that one gene of each pair is present in the gametes, the pollen or the eggs and the sperm. So genetic terms. Gene is a unit of heredity, a section of DNA sequence encoding a single protein. Genome, the entire set of genes in an organism. Alleles, two genes that occupy the same position on homologous chromosomes and that cover the same trait, like flavors, so to speak. Locus, a fixed location on a strand of DNA where a gene or one of its alleles is located. Homozygous, having identical genes, one from each parent, for a particular characteristic. Heterozygous, having two different genes for a particular characteristic. Now let's get into the nitty-gritty. Dominant, the allele of a gene that masks or suppresses the expression of an alternate allele, the trait that appears in a heterozygous condition. So, for example, the brown-eyed allele of the eye color gene is dominant over the blue allele. My mother had blue eyes, which means she must have carried two blue-eyed alleles, because if it was a brown-eyed allele, it would have been dominant over the blue one, and she would not have had blue eyes. My father had brown eyes. I have hazel eyes. So it's logical that from my mother, I must have inherited a blue-eyed allele, and it's masked by my father's allele. But I could have blue-eyed children if my other half is carrying a blue-eyed allele, and a child inherits both my blue-eyed allele and her blue-eyed allele. So dominant alleles are dominant and mask the characteristics of recessive alleles. You need two recessive alleles to have the trait, to exhibit the trait phenotypically. So recessive, an allele that is masked by a dominant allele, does not appear in the heterozygous condition, only in homozygous. So genotype, the genetic makeup of an organism. Phenotype, the physical appearance of an organism. The genotype plus the environment results in the phenotype. For example, perhaps you have a gene that makes you more likely to be tall, to grow tall. But if in your environment you have poor nutrition and you grow up with reduced diet, you're unlikely to be as tall as you would have been if the diet was appropriate. So just having a gene for a trait doesn't necessarily mean that you'll have a phenotype for that trait. It just makes it more likely. And how likely depends on the gene and depends on the environment. A monohybrid cross, a genetic cross involving a single pair of genes, one trait, parents differ by a single trait. P equals parental generation. F1, the first filial generation, the offspring from a genetic cross. This will make more sense when I start the Punnett squares. Just try and keep your head around this for a moment. It will become clear. F2, the second filial generation of a genetic cross. So these are the 7 characteristics in P's that Mendel looked at once again. Just a reminder before we start the crosses. So monohybrid cross. So one of the first things that Mendel did was he crossed a tall pea plant with a short pea plant, purebred tall with purebred short. Now, when a gene is dominant, we represent it with a capital letter. So the capital T here represents the tall gene, and the lowercase t, the short gene. Now, obviously, you have two copies of each gene from your parents. So the tall plant, which was purebred, must carry two tall alleles. So it's got two sets of chromosomes, and each one has a gene for height, and they're both the capital T for tall. The short plant, however, carries two alleles for short. So the combination of the two, if we cross them, would be big T by little t, little t. So parents differ in a monohybrid cross by a single trait, crossing two plants that differ in stem size, one tall, one short. Capital T for tall, lowercase for dwarf. TT, homozygous tall, little t, homozygous dwarf. So this is what Mendel saw when he did this. So he basically crossed the two together. Now remember, when you create a gamete, you only have one copy of each gene. So basically, from the tall plant, you're only going to get gametes with the big T, the gene for being tall. From the short plant, you're only going to get a little t, a single copy of that for being short. The possible combinations in the next generation are therefore going to be big T's against little t's. So every organism, every offspring is going to have a big T and a little t, which means it's going to be tall. Because remember, the big T is dominant over the dwarf little t. This will make more sense as we move along. So, parentals, true breeding, homozygous plants. F1 generation is heterozygous. All offspring will be a big T and a little t, and therefore the phenotype will all be tall. Punnett squares. This is the real clever bit of Mendelian genetics. So Punnett squares can be used to determine the genetic combinations that might result from a genetic cross. The letters in a Punnett square represent alleles. Capital letters, as I've already said, is dominant alleles. Lowercase is recessive alleles. So this square shows all the possible combinations you could have in an offspring if both parents had a dominant and a recessive allele, a big G and a little g, for example. You could get two bigs, or you could get two littles. So you could get a combination of one with two big, one with two littles, and two with a big and a little. But remember, the big G is dominant, so this means that out of each 4 offspring, on average, you would have 3 with the dominant trait showing, 1 with the recessive trait showing. So first, to use a Punnett square, you determine the genotypes of the parent organism. You write down your cross mating and you draw a Punnett square. So for example, if we have 2 big T's, 2 little t's, then we're going to get, let's just jump to it, big T, little t, you make a square. These are the possible combinations for the offspring. So the possible combinations that we have, let me just adjust this chair, there's a bit of sun shining through the window, as we can see on my arm. Okay, so the combinations here, if we mated a purebred tall and a purebred short, then all the offspring can only be big T, little t, which means basically genotypes will be 100% big T, little t. The phenotypes will be at 100% tall plants. So the F2 generation. If you let the F1 generation self-fertilize, the next monohybrid cross would be big T's against big T with a little t against a big T with a little t. Phenotype all tall. And you would get this. So a quarter of the second generation of the cross would contain 2 big T's, a quarter would contain 2 little t's, and the other half would be big T, little t. So genotypes, 1 TT, 2 big T, little t, 1, 2 little t's. So 1 tall, 1 pure tall, 2 heterozygous tall, 1 homozygous dwarf, The genotype ratio would be 1 to 1. The phenotype, 3 tall, 1 dwarf. Phenotype ratio, 3 to 1. So ratios and Punnett squares. Punnett square basically predicts all the possible combinations for alleles for each trait examined. This can be used to predict the likelihood of traits of an offspring, the probability of things happening. It also reveals the ratio of one type of offspring compared to the other types. Ratio can help compare genotypes to other genotypes or phenotypes to other phenotypes. It ultimately shows either possible offspring compared to the other possible offspring or offsprings. Secret of a Punnett square. The key is this: determine the gametes of each parent. How? By splitting the genotypes of each parent, making purebred populations. If this is your cross, the gametes are, so the gametes being the eggs or the sperm, if this was an animal. So all the eggs, all the sperms could only from one side be big T's and from the other side, the other parent, be little t's. And therefore the combinations can only be big T, little t. There is a shortcut. Because you've got duplicates, you can, as you would do with algebra, you can basically get rid of the duplicate. If either parent is homozygous, you know that in terms of the gametes, in this case, you can only get one big T, one little t. So really that square is one big T, little t. You don't need to draw the full 4 squares. Genotypes, 100% big T, little t. Phenotypes, 100% tall. You only need one box, basically. You don't need to use a group of boxes. I think you understand the shortcut, but just to summarize it here, this equals this. Genotypes, 100%, phenotypes, 100%. If you have another cross, however, a heterozygous with a homozygous, so for example, big T, little t, mating with an organism that is 2 little t's, you can still use the shortcut, just not quite as short. So there we go. We know we can get the big T or the little t gametes from one organism, the heterozygous, and from the homozygous, we know we can only get the little t. So this is our shortcut Punnett square. Genotypes, 50% big T, little t, 50% 2 little t's. Phenotypes, 50% tall, 50% dwarf, because remember, the big T is dominant, so anything with a big T is going to be tall. Another example, flower color. So the pea plants can be purple or they can be white. The white isn't showing so clearly here, but take my word for it. So big P is purple, little p is white. So the big P is dominant, the little p is recessive. If you cross a homozygous purple big P with a homozygous white little p, then you get big P, little p in all of the offspring, all purple. So with the first generation cross, the first F1 cross, if you self-pollinate again, so if you cross the next generation, then you get big P, little p by big P, little p, which is going to give you this. 1/4 are going to be white because they've got 2 little p's. The rest are going to be purple because they all have a big P. But some will be heterozygous, some will be homozygous. So the genotypes, 1 big P, 2 big P, 1 little p. Phenotypes, 3 purple, 1 white. So Mendel then came up with his principles based on this. The first principle of dominance. One allele masked another. So one allele was dominant over the other in the F1 generation. So Mendel came up with this idea of dominance and recessiveness. 2, the principle of segregation. When gametes are formed, the pairs of hereditary factors, the genes, become separated, so that each sex cell, an egg or sperm, receives only one kind of gene. So genes and dominance, what is a trait? First of all, a concept you kind of need to get your head around. A trait is a characteristic such as eye color, or such as a particular tendency towards aggression, or towards depression, or anxiety, or schizophrenia, or extroversion or introversion, or being tall or being short. All of these things are traits. Being lactose tolerant or lactose intolerant, they're all traits. An example, tall versus short, so height. Round versus wrinkle, P shape. Human case, cystic fibrosis. So Mendel's principle of heredity apply universally to all organisms. Cystic fibrosis is a lethal genetic disease affecting Caucasians. So certain genetic conditions can affect particular races. So for example, cystic fibrosis affects Caucasians, whereas sickle cell anemia, a blood disorder, affects people of African descent. It's caused by a mutant recessive gene carried by 1 in 20 people of European descent. 1 in 400 Caucasian couples will be both carriers of cystic fibrosis. 1 in 4 children will have it. So cystic fibrosis disease affects transporting tissues. Mucus is accumulated in the lungs, causing infections, causing lung damage, eventually causing lung failure. Inheritance pattern of cystic fibrosis. If 2 parents carry the recessive gene of cystic fibrosis, lowercase c, that is, they are heterozygous big C, little c, 1 in 4 of their children is expected to be homozygous for cystic fibrosis and have the disease. So if you do the maths, that's actually quite low. Even though 1 in 20 of us are carriers, the likelihood of 2 carriers mating and producing offspring that are double recessive, because remember only a quarter of their offspring would both have the recessive allele, would have 2 copies of the recessive allele, is quite low. So big C is normal, big C, little c carrier, no symptoms, 2 little c's, cystic fibrosis. So this is your Punnett square. As you can see, only a quarter of the offspring will have the phenotype of cystic fibrosis. So if we look at probabilities, the 1 in 4 probability of getting the disease is just an expectation, and in reality, any 2 carriers may have normal children. You could have 4 boys in a row, 4 girls in a row. It's flipping a coin. Sometimes you get 4 heads, sometimes you get 4 tails. If you flip that coin 100 times, you're probably going to get about half and half, more or less. It's an important factor when prospective parents are concerned about the chance of having affected children. There are certain conditions which are prevalent, certain alleles which have a higher frequency in certain populations. For example, Mediterranean fever, a blood disorder, is quite common. It's a recessive disorder. It is quite common in people from Cyprus, for example. Now, 1 in 29 Americans is a symptomless carrier of cystic fibrosis. Gaucher disease is another one. It's a rare genetic disease. It causes lipid storage, fat storage disorder. These fats accumulate in the spleen, liver, and bone marrow and cause disease. It's the most common genetic disease affecting Jewish people of Eastern European ancestry. So 1 in 500 incidents. Rest of the population is about 1 in 100,000. So that's the breakdown of Gaucher. Let's talk about dihybrid crosses now. Matings would involve parents that differ in 2 genes, 2 independent traits. We've been working with monohybrid crosses at the moment. So for example, height. Dihybrid is 2 traits. So for example, flower color, big P dominant, little p recessive, and stem length, big T for tall, little t for short. So now we're crossing these traits and we end up with 2 big T's, 2 big P's against 2 little t's, 2 little p's. Possible gametes for the parents are the following. These are the combinations. Now straight away, if you remember the trick for Punnett squares, we can reduce this. The F1 generation, all tall purple flowers. Big T, little t, big P, little p. So using the shortcut, we get just this, much simpler. Dihybrid cross, F2, if F1 generation is allowed to self-pollinate, Mendel observed 4 phenotypes. These are all the possibilities. So 4 phenotypes observed would be tall purple, 9, tall white, 3, short purple, 3, short white, 1. And you can see what the genotypes are. So the phenotype ratio is 9 to 3 to 3 to 1. I'll leave that there for a second, just for you to have a quick scan. Okay. Moving on. So Dihybrid cross, 9 genotypes. Here we go. Genotype ratios against phenotype ratios. So there we can see all the possibilities. We used to have to draw these crosses out when I was doing my biomedical science degree in the first year. Very long and tedious, although the software that does it for you now. This was before the software was around. Principle of independent assortment. So, based on these results, Mendel postulated the principle of independent assortment. Members of one gene pair segregate independently from one other gene pair during gamete formation. Genes get shuffled. These many combinations are one of the advantages of sexual reproduction. Our offspring are different than the parents, and as a result, either cope better or worse with the particular environment that they find themselves in, which therefore can stimulate evolution, evolutionary process. Relation of gene segregation to meiosis. So meiosis is when you make eggs, when you make sperm. So you only get one set of chromosomes following meiosis, following the manufacture of sperm or the manufacture of eggs. The parent organism obviously has got a full set, 2 sets of chromosomes in the adult cells. So for example, all the cells in my body now are what we call diploid, 2 sets of chromosomes, apart from my sperm, which has got 1 set of chromosomes. So this is really what happens during meiosis. Alleles segregate, they separate. So test crosses. When you have an individual with an unknown genotype, you perform a test cross. Test cross with a homozygous recessive individual. For example, a plant with purple flowers can either be big P or little p, big P. Therefore, you cross the plant with a little p, white flowers, homozygous recessive. So big P question, I don't know, does it have a little p or does it have 2 big P's? I don't know because it's purple, could be either. If I cross it with a pure white, then if you get all 100% purple flowers, then the unknown parent was big P, big P. If you get 50% white, 50% purple, then the unknown parent was big P, little p. You get that, looking at the Punnett squares? So you basically make the organisms, you count the phenotypes, you create a Punnett square, you work out the genotypes. Dihybrid test cross. If you had a tall purple plant, how would you know what genotype it is? You cross. You cross the organisms and you cross the offspring. You repeat the crosses, you count the phenotypes, and then you can work out the genotype. So beyond Mendelian genetics, incomplete dominance. Mendel was lucky. Traits he chose in the pea plant showed up very clearly. One allele was dominant over another, so phenotypes were easy to recognize, but this isn't always the case. Not all genes are dominant or recessive. Some have incomplete dominance. So, for example, the snapdragon. Snapdragon flowers come in many colors, very pretty. If you cross a red snapdragon, big R, with a white snapdragon, little r, you get pink flowers. As such. So this is an example of incomplete dominance. You only get red when you've got the double copy of the big R. If you get the big R and the little r, you get pink. Genes show incomplete dominance when the heterozygous phenotype is intermediate. So when F1 generation, all pink flowers, is self-pollinated, the F2 generation is 1, 2, 1. Red, pink, white. Ta-da. So if you get a Punnett square like this, you know you're dealing with incomplete dominance. And there are the possible offsprings that you get from that. What happens if you cross a pink with a white? Pink with a red, sorry, or pink with a red? I seem to have missed a little bit on that PowerPoint slide, but you get the idea. If you're going to cross, you're going to get a mixture of the phenotypes, you're going to get a mixture of the genotype. So the last bit I want to talk about is something called sex linkage. In humans and other mammals, biological sex is determined by a pair of sex chromosomes, an X and a Y. So we have 2 chromosome 1s, 2 chromosome 2s, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, etc., etc. However, when it comes to the sex chromosomes, we only have either an X and a Y, that's the pair, if we are male, or 2 X's, if we are female. So basically, if you're a male, you only have 1 X chromosome, 1 Y chromosome, which means only 1 set of genes on each of those. They're not the same genes, the genes that you get on an X and a Y, they're different. So a female will have 2 sets of genes because she's got 2 X chromosomes, whereas a male will only have 1 set. Okay, so genes on the X chromosome are said to be X-linked. X-linked genes can have distinctive inheritance patterns because they are present in different numbers in females, XX, and males, XY. X-linked human genetic disorders are much more common in males than in females due to the X-linked inheritance pattern. That sun is following me around the room. I should stop and draw the blind, but then I have to mess around with the light, so we're just going to carry on. X-linked human genetic disorders are much more common in males than in females due to the X-linked inheritance pattern. It makes sense. If you've got a gene that is recessive and you have only 1 chromosome, then if one copy of the chromosome that gene is on, then that gene is going to exhibit its effects. Whereas if you have 2 chromosomes, for example, 2 X's, and you have a recessive on one, a dominant on the other, then the effects of the recessive will be masked by the dominant. When a gene is present on the X chromosome but not on the Y chromosome, it is said to be X-linked. X-linked genes have different inheritance patterns than genes on non-sex chromosomes, the autosomes. That's because these genes are present in different copy numbers in males and females. I've explained this. There's just a bit of text there you can read through yourself if you like, but it basically explains the point that I have just made. And this illustrates the point that I've just made even better. So if we look here, we can see a father and we can see a mother. The mother can be the carrier of a particular X-linked recessive allele. Dominant on X, dominant here. So these 2 parents basically do not have the trait. If we look at the offspring, however, an affected son is far more likely, well, in this case, the only possible combination, than an affected female. Because from the combinations, the only person who could really get the 1 copy of the defective gene and for it to have an effect on phenotype is a son. Okay, moving on with an example. Hemophilia. So hemophilia is a very good example of an X-linked disorder. It's X-linked. Symptoms, prevalence, prognosis. It's not too prevalent, luckily. Used to be fatal. Let's talk about the symptoms. So if you have hemophilia, basically you have a faulty gene, which means that you cannot clot your blood properly. So if you get a cut, you can bleed to death. Now we have clotting factors that we can give patients so that they can survive even if they are hemophilic. But in the past, if you were hemophilic, you would probably die as a child from a minor injury. You would bleed to death. An example of this condition and how it works is very well documented and perhaps was responsible partly for the Russian Revolution. So before Queen Victoria, well, the royals in Europe were constantly marrying each other. You know, you'd marry your cousin, you'd marry your cousin, you'd marry your cousin. Everyone would marry each other's cousins. So you all have the same genes accumulating in one population, a very inbred population, the European royal lineage. And that can have consequences. It means that rare recessive genes can accumulate in this small breeding population. Now, we believe that Queen Victoria, there was a mutation. there was a mutation in one of her eggs that she produced. And this was passed on to her offspring, and through lots of interbreeding matings, the gene accumulated. So the mutation would have occurred within Queen Victoria itself on the X chromosome. So she would carry this and pass it on to the next generation. So as you can see, Princess Alice, she carried this faulty gene, Prince Leopold, and Princess Beatrice, and they all passed these genes, this mutated hemophilia causing gene through several lineages. And as a result of that, males in the royal lineage were more likely to develop hemophilia. And a very good example of that would be this guy here, Alexei of Russia. Now, he was the Prince of Russia. He was, at the time of the Russian Revolution, he was a prince, and he was to be the next Tsar. Although that never was to happen because of the Russian Revolution. Now, Alexei carried this X-linked hemophilia recessive gene, and because he didn't have a Y chromosome, he only had an X chromosome, being a male, he exhibited the phenotype and he had hemophilia. And his mother, she basically, so Alexandra, the mother of Alexei, she did everything she could to try and treat her son. And one of the things she did was she called in a monk called Rasputin. You all have probably heard of Rasputin. And Rasputin became really quite involved in the royal family. And a lot of people in Russia did not like the power that Rasputin seemed to have. And it was part of the precipitation for the Russian Revolution. Obviously, there was a lot more to the Russian Revolution than that, but part of the cause of the Russian Revolution was the influence of this Rasputin on the royal family, which wouldn't have occurred if Alexei had not suffered from hemophilia. Okay, so summary of genetics. We've gone through quite a bit. Let's summarize. Chromosomes carry hereditary info, genes. Chromosomes and genes occur in pairs. New combinations of genes occur in sexual reproduction. Monohybrid crosses, one set of traits, versus dihybrid crosses, different traits. Mendel's principles: dominance, one allele masks another, not always, but sometimes. Segregation, genes become separated in gamete formation. Independent assortment, members of one gene pair segregate independently from one other gene pair during gamete formation. And that's all for now. So thank you very, very much for listening. I hope you've enjoyed today's lecture, and I hope to speak with you again very, very soon.